Good afternoon and welcome to the Midwest Glacial Lakes Partnership webinar on the hidden role of iron in primary productivity in some Midwest glacial lakes. I'm Joe Noner and I'd like to share a little bit of information about the partnership and logistics for the webinar before we get started with our presentation. The Mglp hosts these lake conservation webinars to share science, management, and outreach efforts of interest to our partners. You can keep up to date with our webinars by checking out the webinar page at midwestglaciallakes.org. Webinars in the series will be added to our webpage alongside other webinars of interest, and we've got about 20 webinars going this year. Notifications for the webinars will also be provided through the Mglp's quarterly newsletter, which you can sign up for at our website. If you'd like to share your experience or project results through a webinar like this, please reach out to me, Joe Noner, and my email address is on the screen here, nohnerj@michigan.gov. A little bit about the Mglp for those of you that are unfamiliar with us. We are a partnership of professionals and stakeholders focused on conserving the over 40,000 glacially formed inland lakes across an 8-state region. Our mission is to protect, rehabilitate, and enhance sustainable fish habitats in glacial lakes for the use and enjoyment of current and future generations. The partnership provides science for lake conservation, such as our conservation planner that provides information about every lake in the partnership, outreach and education to stakeholders and professionals through webinars, news releases, Twitter conversations, and sharing of materials related to lake conservation. We also provide funding for science, outreach, and education, and on-the-ground rehabilitation projects or protection projects through our Lakes Conservation Grant, which is incidentally open for proposals right now, which closes at the end of this week. The grant is released every year and provides about $300,000 for projects. A few logistics about today's presentation. Should last about 40 minutes and will hold the remaining time for questions. Participants are going to be muted during the webinar, so if you have any questions, hover your mouse over the screen and you'll see at the bottom there's a Q&A button. Just click on that Q&A button and you can submit a question, which I will then moderate and ask to our presenter at the end of the presentation. If for some reason you have technical difficulties or you'd like to share this webinar with somebody else who couldn't attend, we will post a video to the webinars page of our website within a week, and I'll be emailing to follow up with that link once it's posted. Today's speaker is Dr. Betsy Swanner. She's an associate professor at Iowa State University in the Department of Geological and Atmospheric Sciences. She's a biogeochemist who is interested in how iron-rich conditions in aquatic systems influence microbial life through Earth's history. This generally means working in anoxic waters where iron is more abundant and where microbes dominate. Her other favorite elements are those in that cycle with iron in microbial-driven biogeochemical cycles. With that, Betsy, I'll turn it over to you.
Great, thank you so much for the invitation to give a talk today. I'm excited to interact with folks who are also interested in glacial lakes. Let me get my screen working here. So I've been at Iowa State for about 6 years, and I've definitely sort of in that time transitioned from being mostly a lab-based person to someone who's out in the field a lot. And I've come to work on lakes quite a bit and really appreciate having so many sort of in the broader backyard. Before I get started, I do just want to acknowledge that I'm working at a land grant institution, but the land was the ancestral land and territory of the Bikoji or Iowa Nation, and this was obtained from the Meskwaki and Sauk Nations in the Treaty of 1842. And so as Joe Noner already mentioned, I'm a scientist, I'm a geochemist and a geomicrobiologist or biogeochemist or whatever word fragments I decide to put together on that day. And in that capacity, I'm really interested in chemistry, but I'm interested in chemistry that's happening in water. And I'm interested in microbes because microbes are either able to live in different types of chemical environments, or they're able to do different reactions that actually produce kind of crazy chemistry to us. And a lot of these reactions, as Joe mentioned, are happening under anoxic conditions or without oxygen around. And that's allowing a lot of redox or oxidation reduction reactions to happen that change the form of different chemicals like iron and manganese And I'm really fascinated by microbes that can do things like produce this rusty iron mineral, or other microbes that can then breathe it, respire it with organic carbon. And so I'm definitely a bottom-up chemical control type person. That's where my expertise is. I appreciate that this organization is heavily involved in work with fish, and I won't talk about that at all today because I don't know anything about it, but I would love to hear more from you, especially if you have ideas about some of the systems that I work in, what sort of connections there might be with the rest of the ecology. Okay, so just sort of a primer. I'm going to talk a lot about lake chemistry today, and lake chemistry, of course, depends on the availability of elements that are derived, from the substrate a lake is in. But one of the fundamental paradigms that I use as a geochemist and a biogeochemist is sort of this redox cascade. So if we have a system like a lake that is getting organic carbon inputs, either exogenous from the landscape or from in-lake primary productivity, that's providing some fuel, right? And that needs to be respired or combusted or burned, or however you want to think about that, with an electron acceptor. And for us and most other animals, that's oxygen. But the lakes that I'm going to talk about today, there's some limitation on the input of oxygen to the deeper waters, and so they become anoxic. So it's either diffusional limitation or mixing, or consumption, like in a site like this, there's just such high consumption of oxygen that diffusion is not keeping up. And so microbes can then sort of take over and use what other chemicals are present in the lake, maybe salts like sulfate or nitrate, and they can reduce these. So they're essentially respiring using these other chemical components, nitrate or nitrite, manganese oxides, iron oxides, sulfate, or ultimately CO2. And geochemists like to sort of label the different chemistry that results when you have zones of these different reactions. Oops. And the one that I'm particularly interested in is what us geochemists call ferruginous, which is where you have a lot of iron in the system, and because it's being used as a terminal electron acceptor, it's in a soluble form, which I'll talk a little bit more about. When oxygen is gone, iron has the chance to become soluble. My slides are advancing on their own whim here, so I apologize for that. So what I'm showing you in this plot here is that this is some data from a lake that we work on in Minneapolis that has a lot of iron in the system. And you can see that the iron concentrations here with depth. So I'm going to be showing a lot of these plots with depth in the lake. It's about 14 meters deep. There's a lot of dissolved iron in the bottom, but where that meets and interface with oxygen, the iron disappears because the two react. And so we can sustain high concentrations of iron in this lake where the oxygen is gone. And that's going to be really important for some of the questions that I'm asking. And so thinking about this audience of people who are interested in glacial lakes and the glacial history of the area, I want to just sort of do a little bit of a query thinking about iron. Have you ever experienced rusty water, especially if you've been using private or shallow wells? So I don't necessarily have a way to get your answers here, but maybe just think about that for a minute. If you're not on a municipal water supply or have ever been to a place where you're using a shallow well, do you ever experience this problem of rusty water? And I think a lot of us, I grew up in the Midwest as well. I grew up in Minnesota, and that was quite a common scenario when I was out at places that had their own wells that were drilled into the shallow or glacial aquifers, that there's a lot of dissolved iron where the groundwater is anoxic and where there's sort of a geological or sedimentary supply of iron. And so I've been sort of curious about this and thinking about would we expect then lakes that are in communication with groundwater, would we expect then that iron might be kind of a common constituent of lakes as well, especially where they don't have oxygen. And so what I'm showing here is a map. This is data from the Minnesota Pollution Control Agency that shows all these little triangles are wells that are drilled into glacial or near surface aquifers, shallow quaternary aquifers. And the The colors here are showing something about the iron concentration. So the yellower colors have higher iron concentration in the well water. And there's sort of a spatial pattern here. And so the figure on the right is actually the superficial geology, the most recent, or the glacial deposits that are at the surface in Minnesota. And I'm going to focus on Minnesota because I work there a lot. And what you'll see is that most of the high yellow points are in this green glacial material, which is the Des Moines lobe. So one of the most recent Wisconsin glaciations that came down sort of from the northwest. And we did some statistics on this, and indeed from the data set that we have, there is a statistically significant difference between the iron concentrations that of wells that are underlying the Des Moines lobe versus other glacial lobes. And so we sort of hypothesized that, yes, indeed, the glacial history here can have a really big influence on the chemistry of groundwater. And so the next step then would be, well, does this affect lakes? And we might expect that to happen, to have that interaction, if we can have lakes that have anoxic conditions, right? Because I already told you that in order to sustain concentrations of iron in the dissolved form, you need to have anoxic conditions. And so I get really excited when I hear about lakes that are stratified. And I mean, many of you are obviously familiar with this concept, but you know, there's different types of stratification, could be seasonal or could be somewhat intermittent. And I've been particularly working on one end member of these, the meromictic lakes that are permanently stratified. And so I really love this illustration that was done by an artist of one of the lakes that we were working up on in the Upper Peninsula of Michigan called Canyon Lake, because these top layers of the lake that form during seasonal turnover, so fall and spring, they'll mix, but there's always this deeper part of the lake in a meromictic lake that doesn't mix. And because it doesn't mix, it's isolated from bringing in that oxygen from the surface. And in this case, it's deep enough that there's no oxygen production because it's below the photic zone. So one of the other questions I've been really interested in is, well, in midwestern glacial lakes, like, are meromictic lakes common? Like, you know, if we potentially have a lot of background conditions in groundwater that are carrying iron, and, you know, if we have a lot of lakes that are either intermittently or permanently anoxic, then those are sort of conduits or places where iron could build up. And so what I'm trying to show here is plots of data from lakes that I've found. So these two histograms on the right, it's repeating the data that's cross-plotted over here. I'm sorry, these two histograms on the left repeat the data on the right. But all of these data points, the blue ones are from lakes that are known to be meromictic, and the gray ones are from lakes that are either not meromictic or no one's really assessed that. And what I'm finding is that this metric on the bottom, A0, which is surface area, lakes that are generally under 0.5 kilometer squared, I said that wrong. Meromictic lakes are usually under 0.5 kilometer squared in sort of the temperate region that we live in. There's exceptions, obviously, in the tropics, which maybe I'll mention a little bit later. Whereas this other metric, ZR, which is relative depth, and it's, I did not put the equation up here, but it's something like a ratio between the surface area and the maximum depth. That's likely to be above 4, and you can see there's a few blue dots just below that 4% line. And so sort of what I gleaned from this is, like, generally the meromictic lakes in these temperate regions have a small surface area and they're relatively deep, maybe because, you know, they formed as like an ice block or something like that. It's sort of kettle shaped. And intuitively, I think this makes sense. Like, if you have sort of a deep, small basin, it's really hard to mix versus a broad, flat basin. And this is all like sort of intuitive. I mean, I don't think I'm saying anything that's shocking anyone. But the reason that I was sort of looking at this data is when I go back to trying to answer this question, like, well, would we expect iron-rich lakes to be common? We need to have some idea of, like, well, how would we assess this? Which lakes should we look at? There's obviously thousands of lakes. And so these metrics, relative depth or maximum depth and surface area, are data that's usually readily available from state agencies. And so the idea here in one of the projects that I'm working on is to sort of take that level of data and then acquire any chemical data that might exist, and then kind of go and investigate systems that fit at least the screening criteria. And from what I've gleaned so far from the literature, meromictic lakes aren't particularly common, or at least they're not particularly commonly reported, and particularly the spharuginous kind, the ones that have a lot of dissolved iron. And we'll get to why I think that's important. But what I'm showing you is from what I could glean from the literature, from a lot of like interlibrary loan papers from the 1930s and sort of up to the present. About half of these lakes are in the northern temperate regions, and they seem to be associated with glacial conditions. So again, sort of this idea, like there might be some kind of morphometry that's produced from a glacial origin in combination with, whether there's some of these lakes in Canada or northern Minnesota or other places that are in iron-rich bedrock or else glacial material might provide sort of the right geological substrate to produce iron in lakes. There's many other lakes that aren't glacial origin, and so I'm not going to talk about those. There's some in the tropics, there's some in the rift valleys of Africa. Karst terrain occasionally has these, but these systems aren't particularly well documented. But one of the things that I think is important to consider is that as we have increasing human contamination, particularly road salt, this can cause lakes to become meromictic that weren't before. And so we might be shifting some systems into a state where anoxia becomes kind of a permanent feature. Okay, so with that sort of introduction to the type of lakes and the type of systems that I'm interested in, I want to kind of then tackle two scientific questions. And I said I would talk about primary productivity, so I'm going to focus on that. Number one is just thinking about who are the primary producers in these systems. And number two is, does iron stimulate primary productivity? And so many of you are probably familiar with, all the eukaryotic phytoplankton and cyanobacteria. And I'm a micro person, and I get really excited about bacteria. They're very diverse. And cyanobacteria are just one of the photosynthetic clades. They're the only oxygenic photosynthetic clade of bacteria. But it turns out there's a number of different taxa within bacteria that can do photosynthesis, but they're not making oxygen. They're anoxygenic. And so an example here is these purple bacteria that were pulled out of a water column that had hydrogen sulfide. And these organisms can take hydrogen sulfide and produce sulfate using energy from light to sort of provide the energy for that redox reaction. And so I'm going to tell you a little bit more about Brownie Lake, which I sort of already introduced, and our investigation then of, well, could we find these anoxygenic photosynthetic bacteria in such a lake where we know there is oxygen disappearing and there's sort of the right habitat for these organisms. Brownie Lake has been meromictic since about 1920. It's not a naturally meromictic lake, it's a natural lake, but people messed with it, primarily building a railroad embankment. So this is an aerial view. Here's the railroad that changed the shape of the basin. That was in the late 1800s. And then the lake was lowered by connecting it to Cedar Lake. So if you're from this region, you might be familiar with this system. You can kind of canoe under there now, but that catastrophically dropped the lake level in the 1910s. And now there's a ton of road salt. This is adjacent to 394. There's 6 storm sewers dumping into the lake. And so the water column is really stably stratified now. Okay, so I'm a microbiologist, so we can't just easily like go and, I don't know, take a plankton net and look under the microscope and figure out who's there, because a lot of microbes look the same. There's like nothing really distinguishing about them. And so we need to use different tools to assess who's there and what they're doing. And so a lot of times what we're doing is actually sequencing their DNA. So we go to the lake, we take samples from different depths across the chemical gradients that we see, and then we're sequencing a specific marker gene that all organisms have. And in bacteria, it's the 16S rRNA. It's part of the ribosome, or the machinery that makes proteins. And what we found, so this is looking down with depth in Brownie Lake across 3 different time periods during the course of, I believe this was 2017. And what we see is that we have sort of a peak in abundances of purple and green sulfur bacteria. Sorry about that. And this is sort of occurring, I didn't put the chemical plots, but it's like right in that zone where you have oxygen disappearing and iron starting to appear. And so I already introduced the idea that these clades are known often to use hydrogen sulfide, but I haven't told you anything about sulfur in these lakes. I've been talking about iron. And so it's also known that some of these are able to, instead of using hydrogen sulfide, use iron 2 and oxidize it to iron 3 or rust as part of the photosynthetic process. And so we sort of hypothesized that these organisms could be doing that in this lake because there's some sulfide, but there's not really a very reliable supply. And so what we did as microbiologists just took samples and we brought them back to the lab because microbiologists always want to grow something in culture so we can study it. And so we grew these incubations, and from that we were able to isolate an organism. And this is an organism when it grows in these bottles. So, you know, under the microscope, it's tiny. It's not too exciting to look at. But what we do is we put it in these bottles with no oxygen, shine a light on it, and we add the reduced or soluble form of iron. And after a period of a couple of weeks, we see that iron disappear and turn into rust. And here's a control where that is not happening. And so this particular organism that we isolated is a green sulfur bacteria. It's Candidatus chlorobium masuteum. We named that for the Dakota word for iron eating. And this paper is just submitted recently, so hopefully you'll be able to read more about it soon. The other thing that we did is, now that we had this organism in culture and we could tell like it's doing this thing we think might be happening in the lake because the conditions are right for it, but what is sort of the genetic capacity for that? And so the other thing that we did, in addition to just sequencing that snippet of DNA to figure out who it was, we actually sequenced the whole genome. And so this little cartoon here is describing the types of genetic capacity it has. And in fact, it has this particular gene that encodes a protein, Cyc2, that's used by other related organisms to oxidize iron 2. So in fact, we found, it does at the genetic level have this capacity. It can do it in the lab. It can fix CO2 using the reverse TCA cycle. And the other thing we found is that, like, even though this one is related to the sulfide oxidizing bacteria, it doesn't do that. It doesn't have the genes for it. So that helps us sort of pinpoint, like, it must be using iron or maybe a few other things like acetate or hydrogen in this lake. But of course, that's great that we isolated something from this lake and we can show it does something in the lab. But what we're trying to do now is show that these organisms are in fact not only present in the lake, but they also have the genetic capacity as a population to do this thing. And yes, they are in fact doing it. And so one of the ways that we've done that is to do something called metagenomes. So like I said, we sequence the genome of our isolate, but we can also go into the lake and extract DNA directly from water samples. And as I like to say, sequence the crap out of it. So we just sequence like all of the fragments of DNA that we can, and then those can be assembled using computational biology stuff I don't really know the guts of, but what we can do is then assemble the genomes. And so what I'm showing you here in this plot is, again, that abundance of these organisms throughout the water column. They're concentrated at the interface where iron and oxygen meet. And from these 5 different depths at different time points, what I'm showing you here is anywhere there's a star is a unique genome that we sequenced from these lakes. And the color trend of the band is showing how abundant it was at each depth. So we recovered 6 unique genomes that are closely related to our isolate, Chlorobium masuteum. And they all, in fact, do have this pathway, this Cyc2 pathway for iron 2 oxidation, and they lack the pathways for sulfide oxidation. So we're inferring that, yes, in fact, there is a population of organisms that are concentrated in this environment that can do iron-based photosynthesis. What we're working on now is we've tried a couple of different incubations, and we're still working on the protocol. We're actually going to deploy another set of experiments next week where we actually incubate water in bottles from these depths and try to track carbon fixation with and without an inhibitor of photosystem 2, which is the photosystem that oxygenic phototrophs like cyanobacteria use. So from doing that, we're hoping that we can really get a handle on how much carbon fixation is being done by these specific organisms. And so that's work my current master's student, Kaylee Block, is doing, and we'll be out on Brownie Lake next week. I'm excited, hoping for good weather. In sort of a related project, I have another PhD student, Zach, who is going to be, again, working with the same organism in the lab, but also in the lake. So now what I'm plotting from Brownie Lake, in addition to that iron and oxygen data, is the nitrogen to phosphorus ratios. And we still really see the nitrogen, inorganic nitrogen species drop at this interface because nitrogen can also be used as an electron acceptor. And so this organism, it turns out, also has the capacity to fix nitrogen, like a lot of the diazotrophic cyanobacteria. And so we're hoping to then be able to measure rates of nitrogen fixation by this organism in the lab and in the field as well. So Zach will be doing that work as part of a NASA interdisciplinary consortium on astrobiology research grant we have. Okay, so I'm going to take a water break, catch my breath, and then we'll go on to the sort of this last science question. So who are the primary producers? I've told you about maybe this primary producer. You didn't expect these anoxygenic photosynthetic bacteria, but we also have like all the regular players, right? Like all of the normal phytoplankton. And so one of the questions that I've been really interested in is, as you go deeper into these lakes and you start to get into a zone where there's iron, if there's still light available, what is that iron doing to photosynthesis by these oxygenic phototrophs? Are they still active when there's no oxygen? Are they somehow stimulated by iron? Because iron, in fact, is a micronutrient. You don't need nearly as much of it, we or phytoplankton, as nitrogen and phosphorus, but they still have a demand. And it's sort of a differential demand depending on what type of organism you are. So for instance, diazotrophic cyanobacteria have higher iron requirements. And one of the things that we routinely notice, and it's well documented, of course, in stratified ocean regions and stratified lakes, is a subsurface chlorophyll layer, sometimes called a deep chlorophyll maximum. And so this is data again from Brownie Lake. You're seeing the oxygen in the blue and sort of that purpley gray color, iron concentrations. And there's a deep peak of chlorophyll. And what I find really interesting is that chlorophyll layer is overlapping this zone, especially at the bottom of the chlorophyll peak, where you start to see iron concentrations increase. And a lot of these chlorophyll maximum in the literature, when they're in these stratified systems, a lot of times the research is suggesting that there's a benefit in terms of nutrient availability at these interfaces. So I'm curious if iron is also a nutrient that these organisms are benefiting from its enhanced concentrations. So I'm going to be showing you some data from different lakes. We're using a couple different techniques. I'll go back to some of the sequencing techniques again, but this is data from a multi-wavelength fluorometer. So, you know, if you're familiar with just a chlorophyll probe or a chlorophyll sensor, same idea, except now we can use multiple wavelengths. And so we can use the fluorescence from chlorophyll along with accessory pigments to distinguish which class of phytoplankton the chlorophyll A is coming from. So this is just showing that there's a lot more complexity within that chlorophyll A peak of different organisms being abundant at different depths. I mentioned already that we see this in Brownie Lake, and we see it in pretty much every other lake that we've looked at that has iron or is ferruginous. So I'm also just showing you from some data from Deming Lake, which is a lake we're working on. It's up in Itasca State Park, so the headwaters of the Mississippi area. And I find this site really interesting because there's actually a literature from the 1960s showing that there's sort of this plate of cyanobacteria across the lake at a certain depth, 5 to 6 meters, which is also sort of corresponding to that iron-oxygen interface, and it's still a sunlit zone. And we've gone out now, this is our data on the right with the multi-wavelength fluorometer, showing that yes, in fact, like we see also a peak of cyanobacteria similar to what Baker and Brooks saw in the 60s. So I have sort of bits of data sets about the glacial lakes, but I have more of a complete story about a non-glacial lake. And so I just want to walk you through that because I think it tells a little bit about what kinds of questions we're interested in. And it's just a more complete data set. But I'm going to take you away from glaciated environments to northern Germany, to a lake called the Großes Heiliges Meer. If you speak German, it's a large holy lake or holy sea. It's actually a sinkhole lake. So there's some salt topography or salt geology, tectonics below that area. And so this lake collapsed, or the sinkhole collapsed, I want to say like 1,000 years ago, and it was at the site of a monastery. So that's where the name comes from. But I have been curious about this lake because it's dimictic, right? So it mixes twice a year, but it is also ferruginous. And there's iron-rich groundwater in the area as well. And what is interesting about this system is there's a huge chlorophyll maximum in that lake as well, but it is persistent across the sort of summer season. And so in the spring, the sort of salmon colored data points, we see the chlorophyll maximum, and it's well above the position in the lake where you have an interaction between iron and oxygen, right? So that interface is kind of deep at the beginning, the onset of stratification, and then it rises as the lake begins to stratify through the summer until it overlaps with the chlorophyll peak. So in my mind, this lake is a great site to test or to ask the question, like, does iron somehow influence what organisms we're seeing, and whether does it influence primary productivity somehow? And so one of the first things we wanted to know is, like, is it a biomass maximum? Is the chlorophyll maximum somehow associated with an enhanced amount of cells? And so what we did is we just simply counted the amount of cells in the sample. And so in salmon colors in May, when the chlorophyll maximum was huge and iron was way lower than the chlorophyll maximum, we don't see really any difference in cell numbers throughout the water column. It's relatively invariant. But we do see a lot more of a difference, and we see more of a peak at the chlorophyll maximum in cell numbers when iron has sort of come up and is now overlapping that chlorophyll zone. The other thing that I'm showing you over here on the right is a plot of something called most probable number. So that's actually just a cultivation-based technique where we took water from the field and we incubated it in the lab under conditions that would cultivate cyanobacteria. So we're essentially just quantifying cultivatable cyanobacteria. And we see that at both sampling dates, there is an enhancement in the number of cultivatable cyanobacteria in the chlorophyll maximum. It's a little bit higher numbers in the fall. And the reason I think that is significant is because cyanobacteria, I already told you, the diazotrophs need a lot of iron, but cyanobacteria in general have higher iron demands than other eukaryotic phytoplankton. So potentially, this is sort of an indicator that cyanobacteria might be living in the zone where there's iron. Okay, so the other question, or following up other data about biomass abundance, we did a few other techniques here. One of them was, again, sequencing snippets of genes. And for the non-bacteria, the eukaryotes, we have to use a slightly different gene, the 18S rRNA. It's still a ribosome and codes for a ribosome. And so I'm showing that data actually over here. And this is the relative abundance of sequences from different types of eukaryotic phytoplankton in the water column. And what I think is really striking, this is from September, where that chlorophyll maximum is here at around 6 meters. Within that chlorophyll maximum, there's suddenly this spike of green algae that weren't really abundant in the rest of the lake. And data over on the left is from our multi-wavelength fluorometer. And we essentially see the same signal that there's not much of a signal from green algae in the rest of the lake, but where there's this enhancement of chlorophyll, a good portion of that is due to the green algae. And this plot in the middle is showing something called with depth photosynthetic yield. And the green dot there is the yield that is from the green algae. And it's high, it's near the theoretical maximum, which is about 0.8. And this yield is just telling us something about sort of like the efficiency of photosynthesis. And so basically it's telling us that those green algae could have a capacity to do photosynthesis pretty efficiently. So these data are sort of indicators. There's nothing like, proving that cyanobacteria or green algae are deriving like a clear benefit from iron, but they're sort of in the right place at the right time. And unfortunately, with this single study, we don't really have a lot of statistical power to test that, but we do have some ideas now we're taking back to the lakes that we're studying here about how we can better explore that relationship. And sort of the last thing, so I'm going to wrap up here. I said 40 minutes. I think I'm going to be there on the nose, is those indicators of productivity. And so here's again the chlorophyll peaks, and that May chlorophyll peak that happens in the spring when iron is like way down here below it. It's actually completely offset spatially from other indicators of primary productivity. So there's a lot of chlorophyll there, but the peak in productivity seems to be happening higher in the water column at about 4 meters. We see that both in dissolved oxygen is above air saturation at this depth of 4 meters, implying there's a lot of oxygenic photosynthesis. pH peaks, which is an indication that organisms are taking up dissolved inorganic carbon, particularly bicarbonate, and excreting or leaving behind a higher pH environment. And also this slight blip or excursion towards more positive delta 13C of dissolved inorganic carbon, which is indicating that photosynthetic organisms are preferentially taking up that lighter carbon isotope and leaving behind the heavy isotope in the DIC pool, which is also an indication of primary productivity. Those signals are either not there or they're much more subtle when in the fall, when we have a much smaller chlorophyll maximum, but it is still overlapping the iron and oxygen interface. But I think what is sort of interesting is that we do see sort of this slight blip in the blue data points as an indicator of primary productivity occurring with this delta 13 of DIC. So that to me, again, gives us some fine tunes or questions so we can follow up in the future as we go back and start to perform these studies in the lakes in the Midwest. Okay, so I'm gonna wrap up with my conclusions here. Things I want you to take away: iron and meromixis may be more common in the Midwest than previously recognized, and I think that has something to do with the glacial origins. But I think, part of the reason maybe we don't know so much about these lakes is because they're small, and so they're not, the ones that people are like doing a ton of recreation on. And also, generally in monitoring programs, who's measuring iron? Like maybe occasionally that gets measured, but it's not really something that's on the radar, which makes a good hole for us to fill in terms of our research program. Number two, that iron 2 dependent anoxygenic photosynthesis is likely occurring in these types of lakes, and we'll hopefully be sort of quantifying that with some of our incubations that we're doing to say, like, what fraction of photosynthesis could this support in these lakes. And finally, some indications from the initial study that iron may stimulate some types of photosynthesis, and we have an indication from this study in the Grosses Heiliges Meer that cyanobacteria or green algae might be some organisms to follow up on. With that, I'm just going to sort of glide past these references, and I can come back to that in case you want to find any of those. And then I, of course, need to acknowledge a lot of the students, graduate students and undergraduate students that I work with at Iowa State University, collaborators from Minnesota State, University of Minnesota Duluth, and then some collaborators from Germany. And the projects that I talked about were supported by the NSF, a Carl Zeiss Foundation grant, and my startup. And we worked with access through the Minneapolis Parks and Recreation Board, the DNR in Minnesota, and this Landschaftsverband Westfalen-Lippe. I'm not going to say too much more about that one. But thank you all for your attention, and I hope I can take some questions.
Excellent. Well, thank you, Betsy. That was really fascinating, and for a fish person, you know, really sparked a lot of ideas and was interesting for me. So thank you. I think that I speak for those that are in attendance as well. Of course, we can't all clap and applaud you, but I think that folks would if they could. So thank you. At this point, we do have time for questions. Again, if you'd like to ask a question, please hover your mouse over the Q&A button at the bottom of the screen, and you can submit your question in that way, and I'll moderate those questions and forward them on. So the first question, I've got a handful of questions myself. So the first question, Noah, is are you aware of the ontogeny for Canyon Lake in Michigan? You know, how was it formed? Was it a rift lake or a glacial outwash lake?
Okay, so I feel like I need a word definition on ontogeny. Is that origins?
Yes.
Okay. Yeah, so let's see, that was by, I can't remember the fellow's name who just asked that, but we have been working on that lake for a while, and I don't actually know. So Canyon Lake, it really feels like you're in a canyon. And yeah, there is some speculation that it's a glacial outwash, from my understanding. I think we've always sort of, it's in gneiss, like a Precambrian gneiss, as far as I understand it. And we've always maybe thought like it could just be faulted. The fellow asked if it was rift. I mean, I guess if it's faulted, it could be related to rifting, which, you know, when I think about rifting in the Midwest, it's like the 1 or 1.1 billion year old mid-continent rift system. I don't know specifically, but probably some kind of faulting or tectonics, because it is kind of a stark, beautiful setting.
Okay. Yeah. All right. We got another question on equipment. So I think you're probably reading it. What multi-wavelength pherometer are you using?
Yeah. So great question. Most of the data that I showed was collected with a BBE Mglp fluoroprobe. So, you know, when we had sort of high resolution. We do also use a Waltz phytoplan too, which is something that can be deployed. I mean, you could bring it on the boat, but we don't, because I don't want to drop it. But we actually like have little cuvettes that hold about, you know, 2 or 3 mils. And so we put those in there, and the benefit of that instrument is we can do some of those experiments, like to determine the yield or light curves or things like that. So we use both of those. And she also asked, where did we get the working scientist vest? So I ordered those custom. Yeah, I sort of, you know, these discussions about fieldwork safety and especially around safety of students of color. And we do work in Minneapolis, and, you know, I've had the police called on my students on shore. And so I wanted to have something that in that environment they could wear to show like they're really in a professional setting and they're supposed to be there. Great question.
That seems smart. Yeah. Next question. What do you think about the potential role of iron in cyanobacteria blooms in benthic environments or polymictic lakes?
Oh, good question. I'm trying not to think about that anymore. We tried, so I've been in Iowa for about 6 years, and we've tried to do some studies here on iron in lakes, and it's totally baffling to me. I mean, I think there's just a lot else going on in those systems where we don't see like really clear iron signals. We just did publish a paper in Inland Waters about a couple of lakes in Iowa where we kind of monitored a cyanobacteria bloom, and what we observed is like there's like enough iron in the lake to allow the bloom. So, but there is some ideas in the literature that sort of got me interested on this topic too, that cyanobacteria might benefit from the ferrous iron, or what I call this like dissolved form is usually the ferrous or reduced form. And you know, there's a lot of the type of form of iron that cyanobacteria take up and the particular mechanisms that they take up is like still a really active area of research. Some of them can actually reduce iron 3 to iron 2. And you know, they have different transporters. They make these siderophores, these molecules that chelate iron when it's limiting in the environment. And so I think there's probably still a lot of work to be done in these areas, but I'm like realizing I'm not really that great at working at harmful algal blooms, and I want to work on iron-rich lakes. Yeah.
Fair enough. So one of the biggest sources of data that we have across the upper Midwest lakes is citizen science and volunteer monitoring programs. And I'm curious if you've thought about working and collaborating with those, you know, if they provide data that are useful for you, or if you've thought about how you might glean information from them, and either in diagnosing the miromixis or understanding iron dependent dynamics better.
Yeah, that's a great question. I mean, I think one of our project goals at the moment for the NSF grant I have is like just finding more meromictic lakes. And I've sort of proposed not to do it with citizen science, but just with like an army of students, undergraduate students. But I mean, you raise a really good point, because some of the data, like, you know, I sit on my computer and like click through like the state data. I mostly look at Minnesota, and sometimes you find like they've collected either iron, or they are collecting at least like conductivity, and conductivity is really, really important to, you know, in the absence of oxygen data, or maybe it's even more important than oxygen to having a signal for a stratification. And so, yeah, I think that would be great. I probably need to think a little bit more about how to leverage that.
Yeah, okay. Let's see. So one of the other things we're seeing a lot of lately is phoslock treatments and other treatments to lock in phosphorus using alum or other chemicals. And how would that, do you care to speculate on how that might influence the dynamics of the lake, especially with respect to phosphorus or iron?
Yes, I do. So I have done a little bit of a data collection, nothing published, but on a lake in Minneapolis again called McCarran's, which underwent, I think, a series of alum treatments in the past. And that lake was probably holomictic, or certainly it is now, or monomictic, whatever you want to call it. So it's mixing occasionally, but it's stratified, and it probably, you know, it's a little bit of a bigger lake than some of the lakes that I work on. And so maybe it sort of had a propensity to stratify, but that alum treatment, you know, it's adding all this salt, it's adding sulfate.
Mm-hmm.
And so if that does sort of enhance stratification, things go anoxic, and then you start reducing sulfate to sulfide. So you can set up like a crazy, you know, microbial community in those systems. Yeah, so I'm not sure if I answered your question, but in Brownie Lake and in a lot of these meromictic lakes with a lot of iron, there's a ton of phosphate because, you know, when we think about the phosphorus cycle in lakes, a lot of times it's coupled to iron. And so if iron oxides are being formed, but then they're sinking into an anoxic water column and they're being dissolved in the water column and reduced, that's releasing the phosphorus. So a lake like Brownie Lake has really high concentrations. It's got a pretty big reservoir of phosphorus. And so, yeah, I wouldn't recommend like bubbling, you know, that lake and like mixing it all up or something like that as a treatment. I don't know if that answered your question.
No, yeah, I think it does, and I mean, really points to the interconnectivity of all of these different aspects of lake management. And that was going to be one of my next questions too, was about lake aeration, you know, and And so you sort of touched on that. I don't know if you have any additional thoughts on lake aeration as another tactic for managing both nutrients and sediments in lakes.
Yeah, I don't know. I haven't thought about it too much, but the other thing that I would say that I didn't have a chance to talk about today is when you put a lake into a situation where now it's gone anoxic, or maybe it naturally is anoxic because it's meromictic or whatever, a lot of the organic carbon is being decomposed through these terminal electron accepting processes, respiration of iron and sulfate and whatever else is available. But in a fresh water, you're going to run out, and you're going to go into an energetic regime where methanogenesis is favored. And so in Brownie Lake and Canyon Lake and all of these iron-rich lakes where there's data, there's tons of methane down there. And so I think anything that's sort of destabilizing these systems, you're maybe going to be releasing a lot of stuff that you might not want to be releasing.
Yeah, especially given the climate change considerations that we're thinking about now. Hopefully thinking about now. All right, the next question, we've got a question from Ralph. The iron mining pit lakes are typically meromictic. Have you studied any of these?
The short answer is no. Because I've been really interested, when I started this talk about sort of the glacial, like trying to sort of step back and say, like, what is it about this region that might produce a certain type of system, the kind that I'm interested in with iron and stuff. And so I really wanted to approach that of like, what is sort of the natural capacity, whereas a pit lake is like obviously artificial, it has a particular shape because it's been excavated. It has a particular dissolved load because of, this runoff and contamination. The other thing that could be an influence there is buffering, right? Like a lot of these more, or natural lakes would have whatever buffer system they have, usually around here it's carbonate. And in a mining lake, if you have any kind of sulfides or things like that that are weathering, then you're kind of going into maybe a lower pH acidic lake. And I mean, I didn't go into all of this, but like the reason I got interested in these lakes is because they're potential analogs to past oceans that were stratified and had dissolved iron in the bottom. And so like actually some of the iron formations in northern Minnesota, of around like 1.8 billion years ago, were probably produced from a setting like this. So there's like this cool circle of research here. And in those systems, because I was interested in studying those, they're not acidic, right? They're marine waters that are carbonate buffered systems. And so I just haven't been particularly interested in acidic or pit mining lakes.
Gotcha. Yeah, and you alluded to the differences in the chemistry of some of these naturally formed lakes versus some of the unnaturally formed lakes, and maybe even the lakes that are meromictic for unnatural reasons, as opposed to setting up from a more natural perspective. And do you see differences in the microbial communities based on what the driver for that meromictics is?
That's a good question. Yeah, I'm trying to think, like we have a lot of, the microbial data sets get complicated really fast, right? Because you're getting like thousands and thousands of reads. And so we've sort of been looking at it with specific questions in mind. But, we might see sort of different populations of methanogens. Let's take methanogens for an example. Some, they consume hydrogen and CO2, and others are reformulating acetate. And so those are really different populations. And it might depend on like the trophic status of the lake, like what are the fermentation byproducts that are being released. The other thing with the mining in the pit lakes is if you're in a situation, especially in an area that has sulfide, and you're producing a lot of sulfate from oxidation of that sulfide, then that gets into a lake and then it can be re-reduced. And so that will then provide a substrate for these anoxygenic photosynthetic bacteria, but they might be the kind that oxidize hydrogen sulfide instead of iron, or you might have both.
All right. Well, thank you. I am not seeing any additional questions in the Q&A, so I'll take this moment to again say thank you. And we will be posting the video of this webinar to our website, and I'll be emailing a copy of a link to that to everybody that registered. So if you were able to attend, if you got interrupted halfway through and had to take off, whatever the case may be, we'll be able to share it for you and anyone that's interested in it. So again, I'll say thank you. And with that, we will conclude this presentation. Thank you, Betsy.
Thank you all.